Good morning, everyone, and welcome. I'm Margaret Smith, President of Southern Tier Lyme Support Inc.
And I'm Maggie Whitaker, Vice President. We have an exciting day ahead, so thank you for joining us.
We'd like to thank Dr. Gerudo, Amanda Roome, and Binghamton University for co-sponsoring our third annual Lyme Conference. Our organization is a nonprofit whose mission is to educate and spread awareness about Lyme and tick-borne diseases and to provide support for those suffering from it. Education is so important because there are a lot of people getting sick. STLS Inc. is 100% volunteered and relies on the generous support of individuals and businesses to be able to provide a conference of this magnitude. We'd like to take this opportunity to thank our many sponsors and donors and to thank all of our volunteers. This includes our board of directors for their many hours and tireless efforts throughout the year, our support group members, community members, and Binghamton University students. We would like to give a special thank you to Lori Tebby, this year's vendor coordinator, and to Katha Mitchell for her years of commitment as our support group co-chair. We are dedicating this year's conference to Katha's 19-year-old daughter, Kira Mitchell, who passed away this past February due to complications of Lyme and tick-borne disease. The Mitchell family is with us today, volunteering, and our thoughts and prayers go out to them.
A few housekeeping items before we get started. First, if you have any questions or need any information, please don't hesitate to ask. Our volunteers are designated in the lime green t-shirts. If you, at this time, could please either put your cell phones on silent or turn off your ringers, we would appreciate that. The restroom locations are in the entrance hallway towards the back of this room. There's some in the mezzanine off room 2011 and off the rotunda and upstairs near room 2008. Please take rest breaks as needed as we have a very packed schedule today. Feel free to get something to eat at the cafe, or you might want to slip out for a little bit of fresh air. I have a quick word about the question and answer session today. There's boxes. If you haven't done so yet, please use the index cards in the lobby. They can be deposited in the appropriate box for Bob Shagir, Dr. Bach, and Dr. Horowitz. In addition, this year you can also tweet your questions to @STLS Group. I will be presenting as many questions as time allows after the completion of each presentation. If there is additional time, we can open up the microphone to the audience, but we encourage you to take advantage of the cards or Twitter. There are also 3 Lyme quilt panels in the back of the symposium for all of you to view. The red squares represent those fighting Lyme or other tick-borne disease. The green squares represent those individuals that have passed from Lyme or other tick-borne disease.
We would like to invite up Jill Auerbach, Chair of the Hudson Valley Lyme Association, for a brief announcement. Please welcome Jill.
Thank you so much for inviting me to say a few words. I want to give you guys a couple of hopeful messages. We've been working a lot on legislation in New York State. We passed a law a few years ago that allowed our physicians to treat beyond 3 to 4 weeks without being harassed by the government. Senator Serino and Assemblywoman Barrett have passed new laws, including education and such. So we have a lot going on in New York State. Likewise, in the federal government, there was a law passed, 21st Century Cures Act, and through, I can't even begin to tell you what we went through, but we did get a section on Lyme and tick-borne, actually it's called tick-borne disease, which is great. passed. So that is very, very encouraging because it establishes a work group that will have both sides of this issue, scientists, doctors, and even patient reps, et cetera, sitting down at the table together so things aren't done behind closed doors. Additionally, there is a new test coming out that I keep my fingers and toes crossed that will be using something called RNA, which is proof of active organism in our bodies. And it will not only test for just Lyme disease, but basically all the known tick-borne disease pathogens and some others that are misdiagnosed with Lyme. So toes and fingers crossed, that should then validate all the other testing. Will no longer be working in the blind. They'll be able to say, oh, this works for this disease, that works for that. Right now, we don't really know what works because we don't have anything definitive. So that's very, very hopeful. Likewise, this is our county proclamation, Dutchess County. You guys can do this too. If anybody wants to know how to speak with all of their local legislators or their state or federal, come up to me, I'll be glad to help. And this is the New York State proclamation. It was just signed into law on Wednesday. I do have copies of a lot of information that's going on. Just want to say Lyme disease and tick-borne disease costs about $4 billion a year to our government. The CDC only gets $10 million a year for Lyme disease, and the NIH only gets about only spends about $24 million. Something needs to be done, and we're working on appropriations as well. So there's hope, guys. That's it. Thank you.
Now it is my pleasure to introduce Dr. Ralph Gerudo. Dr. Gerudo is a professor of biomedical anthropology at Binghamton University and a member of the National Academy of Sciences. He has worked on, on infectious and chronic diseases in human populations during his tenure at the National Institute of Health and at Binghamton University. Dr. Gerudo has over 400 publications and is working on ecological, spatial, and human dynamics, education for Lyme in places where people live, eat, and recreate in what is referred to as built environments. Please give a warm welcome for Dr. Ralph Gerudo.
Thank you, Maggie and Margaret. This is now our third Lyme conference, and I would like to thank, of course, you, Dennis, and all the volunteers at Southern Tier Lyme Support for all you've done to help the community in the region in dealing with the burden of Lyme disease. Of course, without your efforts and of course, organizations like yours, a number of them here today, we would be much more challenged in dealing with the rising problem of Lyme and other tick-borne diseases in our region. And in many ways, Mandy and I and all of our research team are kind of on the front line, being able to see what is happening even before you folks wind up with the Lyme disease cases that develop. We're in the field looking at the prospects of what will happen in the coming days and months and years ahead. As a segue then into why Lyme disease is rising, I would like to introduce Mandy Roome, who's been working with our Binghamton University Lyme and other tick-borne disease research project for almost 5 years now. And she's been instrumental in the success of the project and has trained a large group of undergraduate and graduate students in Lyme disease research. But she's also been an important part of community efforts and programs to make people aware of Lyme disease, not only in New York State, but in Pennsylvania and in New Jersey. She's familiar to many of you as she has given her time and expertise to help local communities understand the risk of Lyme disease and to help them move forward with mitigation and prevention strategies. Please give Mandy a warm welcome.
Good morning, everybody. If I end up being too quiet, please start waving at me and let me know so I can speak a little louder. Good morning, and thank you everybody for attending this event. Thank you to the Southern Tier Lyme support for doing the hard part of this. Dr. Gerudo and I get off very easy organizing this event, so thank you to you guys, and thank you for everybody attending. And a huge thank you to Physical Facilities for setting up everything for us. This is a very important event. I'm so happy that we're in the third year of it. It's very important to get the scientific research, the clinical research, doctors that are treating patients and patients to come together to talk about this problem so we can move forward and try to make it less of a burden. So what we want to do today is to talk about what we've been up to since we spoke to you last year, or for those of you that haven't heard from us yet. So the first recognition of Lyme disease was in the 1970s, so in Lyme, Connecticut, hence the name. There was a cluster of cases of what was seen as some sort of juvenile arthritis, rheumatoid arthritis. Nobody really knew what was going on. Arthritis is not normal in children. So they saw that this was something very similar to a condition that was already known in Europe, and it was already known as a tick-borne condition. So we then named it Lyme arthritis at the time, and in 1976 we renamed it Lyme disease. So the first cluster of cases, these were the first 52 cases in the 1970s. As you can see, children were disproportionately affected by this, and the black circles talk about the EM rash, the erythema migrans rash. So I know it's the telltale sign of Lyme, but as you can see, even from here, it's not very common. This is about 25% of people showed up with the rash, which means 75% of people didn't. And I'm sure a lot of you can attest for that have had Lyme in the past or currently. So the causative agent of Lyme disease is a bacteria called Borrelia burgdorferi. It was discovered in 1981. The studies were published in 1982 by Dr. William Burgdorfer, and he did this research at the Rocky Mountain Laboratories in Colorado. So the causative agent, it is a tightly coiled spirochete, so it's a bacteria. It is the fastest moving spirochete known to science at this time, so it is very good at evading the immune system, as unfortunately a lot of you probably know all too well. So the primary vector for Borrelia burgdorferi is Ixodes scapularis. We call them the black-legged tick. Most of us know it as the deer tick. So there are 3 stages. So the larval stage is very tiny. I do have ticks at the BU table in the back. They are very dead. They will not move. But the larval stage is very, very tiny. It's about the size of a pinhead. It's nearly impossible to see. They only have 6 legs, so they're a little bit easier to identify. They are not seen as a huge risk. I'm not saying they are no risk to humans. They cannot carry Borrelia burgdorferi. It cannot be transovariously transmitted, so when a female lays her eggs, if she has the Lyme bacteria, she cannot pass it to those eggs. However, there are other strains of Borrelia, so Borrelia miyamotoi can be transovarially transmitted in pretty low levels, so they're still a risk, but they're not a huge risk to human infection. The nymphal stage is the next stage, so once a larva takes a blood meal from a human, from a mouse, from anything, it'll molt into the nymphal stage, and this is seen as the most dangerous stage to humans. These are responsible for about 90 to 95% of cases. They are active in the summer. They'll probably start coming out soon, unfortunately. Maybe already, some of you may have seen some. They are also very tiny. They're about the size of a poppy seed. If anybody's ever seen that wonderful public health ad with ticks on a poppy seed bagel, I don't eat poppy seed bagels anymore. But they are very tiny, and unfortunately they are out in the summer, so that's when we're in t-shirts, we're in shorts, we're outside because it's beautiful. So unfortunately, because they're so tiny and because we have a lot of skin exposure, they are responsible for most human infections, so they are seen as the most dangerous stage. When a nymph takes a blood meal from whatever it feeds off of, they will molt into an adult. At the adult stage, that is when they have morphological differences, so you can tell the difference between the adult males and the adult females. So the adult males are very dark black, they have a pretty rounded head, and the adult females are a little bigger than the males. Usually they're kind of reddish orange on the back, and they have a very pointed head. And the females tend to be a little bit more aggressive than the males, so you probably see them more often. And then the adults will mate. Usually this happens in the fall, so if any of you are hunters or just see deer, Deer are usually covered in ticks. Deer are the primary breeding site for ticks. So they will mate, and then the male will die, and the female will drop off, and she will lay her eggs. She lays about 1,000 eggs at a time. So this is why controlling tick populations is so hard, because even if you have over 90% mortality rate in tick populations, the population of ticks is still growing. So it is very, very hard to keep control of tick populations. So the reservoir host for Borrelia burgdorferi is the white-footed mouse, Paramyscus leucopus. There are other small mammals and birds that are also hosts, but what we focus on is what we call reservoir competency. So that's the ability for a mammal to transmit the bacteria, if they have it, to a tick. So mice are the most competent. About 90% of the time, if a tick is feeding on an infected mouse, it will pick up the bacteria. And the infection is circulated between mice and ticks, so if you have an uninfected mouse and an infected tick bites it, it can give it to that. So it just goes back and forth. It's the gift that keeps on giving. So transmission is, again, like we said, from tick to mouse or some other animal. Ticks give it to us. Unfortunately, mice do not give it to us. It is transmitted during feeding, and I can already see eye rolls. I'm going to get to the time frame in a second. So the tick, when it's feeding, the bacteria is living in the midgut. So when the tick starts to feed, it's taking in red blood cells, and it's returning the plasma back to you. So that process does take a little while. So as the plasma is going back in, the spirochetes are migrating up, and they will go into your body or whatever host it's feeding off of. There are some studies that say that the allotted time, I suppose, is 36 to 48 hours for successful transmission. However, these are lab-controlled studies, so this is not what happens in real life. There is a lot of anecdotal evidence to suggest that this happens a lot faster. If this feeding process had started earlier, so say a tick had bitten me and I picked it off and threw it back in the woods, and then Margaret walked by and the tick got on her, that process already started. So that 36 to 48 hours is gone. So anytime you get bitten by a tick,
Kill it.
just, yes, kill it. And definitely go to the doctor. So just to talk about the magnitude of the problem, I know I'm preaching to the choir here, you are all aware of the problem. I just want to go through some of the maps from the CDC of the number of cases that are in the US. So this is from 2001. These blue dots, they're just dropped randomly in a county from which the case was reported. So this is a map from 2001, 2006, 2008, 2011, 2013, and 2015. So this is a problem, as you can see. To kind of understand the problem, Lyme disease is a national notifiable infectious disease, so doctors are required to report it. If you go by the numbers that are reported, this is from 2013, so 36,000 cases of Lyme disease were reported in the US in 2013. 13,000 of them came from the Northeast. The majority of Lyme cases, almost virtually all cases, come from the Northeast and Upper Midwest. So that is a huge problem. But the CDC in 2013 came out and said that they don't think these numbers are very accurate, and I'm sure a lot of you have heard this. They think that this is vastly underestimated, so they think the true number of cases in the US per year is 300,000. So that's 10 times more than what is actually reported. So Paul Mead, he is the chief of epidemiology and surveillance at the Lyme disease program at the CDC. So he said that they know that the routine surveillance only gives part of the picture. So this is a growing problem, and if we look at the new number, unfortunately Lyme becomes the third most prominent notifiable, national notifiable infectious disease. So it is a big problem. There are a lot of cases. We certainly have to do something about it. So I want to talk about the incidence, the Lyme incidence in New York State. So we are working in the southern tier mostly, so that's the counties that are highlighted in yellow here. And we're also in Otsego County, which is the furthest south county in blue in the Mohawk Valley region. So these 6 counties make up the upper Susquehanna River Basin of New York, and that is the area in which we are working. So if we're looking at the crude incidence rate from 2012 through 2014, the southern tier has about 73 cases per 100,000 population, and Mohawk Valley has about 51. So that seems really low. So if we're looking at the capital region or the Mid-Hudson region, we're in the hundreds. And we'll show you later, the ticks that we're collecting, the infectivity in the ticks is very, very similar to that in the Hudson Valley. So why do we have so fewer, supposedly, Lyme cases than the Hudson Valley? So I also want to bring up county variability. The New York State Department of Health has all of this data on their website. It's publicly available if anybody's interested. So even though we kind of group things into regions, things are very different by county. And even within county, things are very different. So if you're looking at Delaware County, the crude incidence rate is 43 per 100,000. But then if you're looking at Tompkins County, it's more than double that at almost 90. So even if, you know, your area seems to have a low rate or a high rate, look at your county, see what's going on in your county. So I also want to bring up an article. It was published in 2015. Jennifer White, she works for the New York State Department of Health up in Albany. She did it was a couple-year study on underreporting of Lyme disease cases. Because as we said, these numbers that we're looking at are really low. It doesn't really match up with the ticks, with what we're looking at. So they did a study. They wanted to kind of assess what was going on and why we have such low numbers. Is it being underreported, or is this just accurate? So what she found in this study, what her and her team found in this study, was that it seems, and these are her words, not mine, so if there are physicians here, don't get insulted. It seems that reporting Lyme cases gets cumbersome for physicians, so they just stop doing it, unfortunately. And I'm not saying all physicians do this, but this seems to be a big problem that she came across, and also misclassifying in coding is a very huge problem for misdiagnosing Lyme. So as a lot of you I'm sure know, if your patients, former or current, Lyme disease is known as the great imitator. So there's a huge array of symptoms. So a lot of times it is misdiagnosed because there's symptoms that are very common to other infections as well. So unfortunately, between the miscoding and just the misidentification and just not reporting, they found that a minimum of 20% underreporting is happening. This study was done in 3 counties near the capital region, but they believe that this is very similar for the rest of New York. So it is very underreported in this area. So now to get into our project, Dr. Gerudo, who you just heard from before, started this project in 2011. I started working with him in 2012, and the rest is history. So the central hypothesis for our project is that fragmented ecosystems within built environments that have high human foot traffic will create areas where contact with the infectious agent for Lyme is very, very high. So by fragmented ecosystems, we mean, and built environments, we mean areas that are broken up. So we have forested areas that are broken up by roads, buildings, just man-made things. So the perfect example is suburbs, which is what the Northeast is made out of. So our sub-hypotheses is that there is going to be an uneven distribution of infected ticks in high densities in areas where there are more people. And we want to look at the social, behavioral, and demographic variables. We feel that they are very fundamental to mediating risk of infection in humans. Mice don't get sick, ticks don't get sick, so we're very concerned with people, because people, horses, and dogs. I study people, so sorry to the horses and dogs. So we're very concerned with what people are doing and how to mitigate this. And a natural and experimental model and a systems dynamic approach will allow us to develop mathematical models of risk of contact and infection, and we hope to use these in intervention strategies by the public health community. So the objectives of our project are to determine the risk through the tick spatial distribution, density, and pathogen prevalence of risk near to human activity. We want to identify, categorize, and quantify the behavioral, social, and demographic variables of infection risk, and we want to develop this mathematical model. So if I had to, this is interactive now. So if I had to ask you guys where you think the highest prevalence of Lyme in ticks is, where in this picture do you think it would be? Okay. I think so. Sorry, too many people. So I think most of you said this. So yes, and our data is actually showing this. Where you have more people, you have more infected ticks. It's not when you're in the middle of nowhere, you know, backpacking the Appalachian Trail. It's in your own backyard. It's in the county parks that are pretty tiny. All of these areas that you typically wouldn't think are a high-risk area. So the reason that this is an issue if you have what we call an intact forest, so you have an uninterrupted forest, it just goes for miles and miles and miles. There are no roads, there's nothing there. So of course, you still have your ticks, you still have your mice, and you still have some infected ticks. But your numbers are kind of in check. But why is that? So we have predators. We have a high species biodiversity when we're in these areas. So we have foxes, we have coyotes, we have bobcats, and whatever else eats rodents. So it will keep these rodent populations in check. The ticks have a lot of opportunities. They have a lot of options to feed off of. So it's not always going to be the very competent mouse. Generally, it tends to be the larger the mammal, the less competent it is of being a host. So when we have this, we have a happy ecosystem. Everything is in check, everything's kind of kept in balance. So you still have some infected ticks, of course, but they're in check. However, when we create these fragmented forests, so we create these suburbs, which, the Northeast loves suburbs. So we are breaking up these forested areas. So we still have enough of an ecological area to support ticks and mice, but we don't have enough of an area to support the predators anymore. So we now are driving out the predators. We have nothing to keep rodent populations in check. So we are skyrocketing the rodent populations, therefore skyrocketing tick infectivity and density. So our goal is to build a risk model. So all of the variables that have an effect on Lyme disease, we want to take a look at and try to create a model to kind of figure out what exactly is the biggest contributor to people coming in contact with infected ticks. So we collect rodents. So we have an IACUC and DEC approved protocol. So we are certified to collect the white-footed mouse, the meadow vole, the meadow jumping mouse, and short-tailed shrews. These are all competent reservoir hosts. As I said, the white-footed mouse is the most competent, but these others are pretty high as well. So we collect these mice off the trails along the side of high-use foot traffic trails or pathways or walkways. So the animals are sacrificed in the field according to IACUC guidelines. We don't need to get into that part. So we take them back to the lab, and we want to see kind of where the spirochete is hanging out, where this bacteria is in the mice. So we tested a bunch of blood samples, and we wanted to see 1.7% positivity, which doesn't really make sense. So our ticks, which we'll get into in just a couple of minutes, our ticks, 1 in 3 is infected, sometimes a little bit higher than that. So the most competent reservoir having less than 2% infected makes absolutely no sense. So what we wanted to do was start to dissect the rodents and kind of see where the spirochete is sequestering. You'll see a wonderful video, I believe, of the spirochete jumping out of the blood vessel. So the spirochete is known to kind of lodge in other organs. So we started dissecting the mice, and we wanted to see kind of what was happening. So we have the highest infectivity is in the bladder at 35%, but it is really important to note that out of the mice that we've dissected everything, so we have the blood and every single organ, 50% of the mice whose blood tested negative, a different organ tested positive for the Lyme pathogen, which is very interesting. So if you're solely relying on a blood test to determine the prevalence of infection in mice, it's going to be very, very skewed. So we have, in general, I think it's 51.7% of the mice are infected, but if we're just looking at the blood, we're going to say it's less than 2%. So that is really interesting, and I know everybody's looking at the brain right now and seeing 50%, so I do want to talk about that. At this point, we have only dissected 2 brains. So we don't want to say that the prevalence is 50% in the brain. The next 98 we dissect could be negative, we don't know. So hopefully we will have more of an update for you on that next year. And the overall goal, so looking at where this bacteria sequesters, what we're looking at too, Dr. Darcy, for those of you that were here for the first year, he's a former student of Dr. Gerudo's. He has a cohort of 250 chronic Lyme disease patients. So what we would like to do in the future is to see if we can correlate where this spirochete is sequestering and with Dr. Darcy's data or with previously published literature, kind of if there is any potential correlation between what symptoms people are having and where this spirochete is sequestering in the mice. So another part of our study is to look at vegetation. So we're looking at the canopy cover, the understory, the ground cover. So we want the leaf litter depth, the soil depth, the amount of mast, so the amount of acorns that are produced and other nuts, We don't have this data analyzed at this time. We are still working on that. So again, hopefully next year we'll have more for you. So tick collection, we collect them by the dragging method. This is what everybody uses to collect ticks. We use a 1 by 1 meter corduroy cloth that's white, and we drag it along the pathways of high human use. So right along the edges where you could very easily brush up against. We want to see what's going on. So the ticks, the corduroy with the ridges is very easy to grab onto. So the ticks will hop on that drag, and then we kill them. So we make very detailed maps. So we collect the ticks and the rodents in the same places. We want everything to be exactly the same. So we make very detailed maps so we know what trail we're on, we know what side we're on, so we know exactly what's happening and where. So the preliminary data for the tick density, this is data from October of 2012 through August of 2016. So the total area that we've dragged is about equivalent to 126 football fields. So it's a lot of area. So these, again, are all high human use walkways. We're not in the middle of nowhere. We're going where people are. So BU campus is pretty high, so you're probably all gonna leave here itchy, sorry. There's a high density of ticks here. Overall, in the 6-county region of the upper Susquehanna River Basin, there are 3.8 ticks per 1,000 meters squared, which is pretty high. You know, it doesn't seem like a lot, but 1,000 meters squared doesn't mean much to me. So about 1/5 of a football field, less one end zone. That's what 1,000 square meters is. So if you think about how often you walk that in a day, if you're taking your dog for a walk, if you're taking your kids to the park, if you're a student here, if you're just walking from your dorm down to campus, you walk that area a lot. So potentially coming in contact with this many ticks is pretty scary, because the amount of times you walk that, multiply that out, and that's how many ticks you could expect to pass. But let's also talk about how many of them are infected. So we do the lab analysis after we collect everything in the field. So we want to identify the species. We want to make sure we have deer ticks. We do have other ticks here. We're very focused on the deer tick because we're focused on Lyme. The deer tick is the only one that can carry Lyme in the Northeast. We also want to identify the life stage, so we kind of see where our biggest risk is, and if they're adults, we want to note whether they're male or female ticks. So we do DNA extraction, and we do PCR amplification, so we're targeting the outer surface protein C for Borrelia burgdorferi, and we run gel electrophoresis to identify the positives. Any positive products we have, we will Sanger sequence to confirm the pathogen identity. So the preliminary data for our infectivity, so the way this table is set up, the first number is the percent infectivity, and the number after that in the parentheses is the number of ticks that we've tested. So we have a lot of ticks, of course. So BU is pretty high. We have a pretty high infection rate at BU, followed by Delaware County and Tompkins County. It is worth noting we don't have a lot of ticks in Delaware County. I am not saying Lyme is not a risk in Delaware County. If you look at the New York State Department of Health, there are a lot of Lyme cases there. Ticks just don't like us in Delaware. I don't know what to say. So we do have a pretty small number there. And as we would guess, so we've got the total number of ticks infected, and then we have the nymphs, and then we have adults. So the percent of nymphal ticks that are infected is lower than that of adults, which makes perfect sense because the nymphs have only had one chance to take a blood meal, where the adults have had 2. So we would expect the adult infectivity to be much higher. So I do want to talk about the density of infected nymphs. So as I said, the nymphal stage is seen as the most important stage, the most dangerous to human infection. So we want to look at that. A previous study by Dr. Duke Wasser, she identified that a density of infected nymphs of 0.3 per 1,000 square meters is identified as a high-risk area for human infection. So we wanted to figure out what was going on in all of these counties with the nymphs, and it turns out almost all of our counties are at 0.3 or well above, which is a little scary. I do want to note, so Delaware County is lower than that. Again, I'm not saying it's no risk. And Tioga County, for those of you, I just did a talk on Tuesday to some folks. I talked about county variability, but also within county variability. So if we broke it down, which I did for the folks on Tuesday, one park, the DIN is above. It is a high risk area, one park is not. So then when you pool them, it looks like the county is not a high risk. So I guess the takeaway is be careful everywhere. So the most important part of our study is the human demographic and behavioral studies. So we need this to develop the risk model. There are a bunch of risk models out right now, but they're just purely ecological. It doesn't have the human component, and that's what we're worried about. We get sick, that's the problem. So what we do is people watch, I guess. We go to these high foot traffic walkways where we're collecting the rodents, where we're collecting the ticks. So over the course of an 11-hour day, we watch the people that go by. We see what they're doing, we see what they're wearing, what sort of risk they're putting, whether they know or not, but what sort of risk they're putting themselves at when they're out. We also note the garbage cans. We want to see kind of what's in them. So if we have trash cans full of food, if we have food all over the path, obviously that's going to attract rodents. And we've learned that rodents are not helpful in this. So some of the data we have, this is from the fall semester of 2013 on the BU campus. There's a very high risk. Again, you don't really expect on campus that you're gonna potentially, while you're going to class, come in contact with a tick. So clothing choices are vital to expose skin for ticks to kind of climb on and hang on. So overall, if you count any risk at all, over 70% of people were at some sort of risk. And then we just told you there's 42% of the ticks on this campus are infected. So that's a huge number. That's a lot of people at risk. And then switching gears to summer within the 6-county range that we're in. So these are all in state and local parks. Unfortunately, virtually everybody is at risk, unfortunately. You would expect that, when you're going for a walk in the park and it's 85 degrees in the summer, you're not gonna think to wear your long pants, wear your long sleeves. So people are at a very high risk, and unfortunately this is when the nymph stage is active. This is the most dangerous time for human infection, and this is the highest risk time as well. So what is this? This is what we call a causal loop diagram. I know you guys can't read this, and that's completely fine. We're trying to develop this model, so we create causal loop diagrams first to kind of look at the variables that affect Lyme disease outcome. So we've got rodents, we've got awareness, we've got clothing, we've got larval, nymph, adult. We have everything coming in. So those of you that have been affected by Lyme disease or know somebody personally, Lyme is not a one and done. There are so many things that affect it. So we came up with this causal loop diagram of all the things we could think of that kind of play in the outcome of human infection. So we start off with this. This is phase 1 of our model. And then phase 2 of the model makes it a lot cleaner. We create a simulation model. So the blue variables that we have here are ones that we had to calibrate off of the model, and the red ones are data that we collected ourselves. So what we want to do is kind of predict the number of Lyme cases. So we'll run simulation models. So first we started on campus because we have most data is from the campus. And the goal is to now expand this model into the 6-county region of the Upper Susquehanna River Basin. So this is the output of the simulation model. So we looked at historical cases of Lyme disease. So we have matched up pretty well with our simulation model for predicting the number of cases. And we used maximum likelihood estimating approaches. And of course, we do have a lot more to do. Again, this is scaled down to the BU campus, but what we're looking to do is to expand this model into the 6-county region that we've been working in. And then after that, what we want to do is kind of expand and hopefully have a model that can be applicable to the entire Northeast, because it will help with public mitigation strategies for sure. So who wants to make a causal loop diagram? So today, what we're going to do at the end of the conference, after the last speaker at 4:30, we are going to have a focus group meeting if anybody is interested in attending. There's a conference room kind of right when you walk in. When you walk in on the right, if you go this way on the left, there's a sign on the door. But we are asking for your help to develop a model to understand how Lyme continues to progress in our area. The 4 of us that are working on this model, we have been fortunate enough to not have Lyme disease. So there are things we don't know. So we want to talk to patients, both former and current, as well as healthcare providers that treat Lyme patients, to help us to really make a model that makes sense. We have what we think is useful in the model, but we want more. We want to hear from you guys. This session will take about 1.5 to 2 hours. It's just kind of sitting down and chatting with us. You guys will remain anonymous, your participation is voluntary, and you are more than allowed to leave at any time that you choose. So if anybody's interested in that, it is at 4:30 today, and we will remind you a little later on in the conference as well. And also, another thing that is going on, those of you that were here last year may have heard of this, you may have taken it already. I am running a Lyme disease survey to look at the lifestyle impacts of Lyme disease. So I want to see the impact on human health and see how things are changing as a result of Lyme diagnosis. And the ultimate goal of this study is to kind of assist in public health mitigation and prevention strategies. So if anybody is interested in that, I do have copies up at the BU table. I have a link, I can email it to you, you can type it in yourself, however you want to do it. But I do want to talk about some of the results of that survey. So those of you that have taken it last year, I appreciate that. Right now I have 308 participants, so I have about 50% patients, 50% controls. This is a self-reporting survey. 42% of the patients that took this survey have been diagnosed within the last 5 years with Lyme. And 77% of these patients had ongoing symptoms after their initial treatment. So that is a really high number. I know it really doesn't shock those of you that are patients here. 63% of these people still have ongoing symptoms, and these are symptoms that are lasting over 1 year. And more than half of patients, which we would expect, don't remember ever being bitten by a tick because they're so tiny. So exercise and social activity significantly decreased after patients were diagnosed with Lyme disease, which we would expect. Unusual fatigue after a normal night's sleep significantly increased after Lyme diagnosis as well. And what I found really interesting was the prevention and mitigation strategies, as you would expect, did significantly increase after Lyme diagnosis. But what it seems to be with this data is that these prevention and mitigation strategies don't prevent an initial infection. They prevent additional infections, which is not what they're designed to do. So we do have data to see the people that have taken it to see if they know someone with Lyme disease and what degree of relation they are. So what we want to see is what it takes to get people to take these precautions. Does it take a direct relative? Does it take a friend? What does it do? So this is something we want to do in the future. If anybody is interested in participating, it is still open for a couple more months. So come see me, come to the Lyme table, and you can do that. So just 2 more small projects, finally something happy. We are doing a small project on household pets as sentinel species. So basically your dog will go outside, they'll pick up a tick, and we love our animals, so we let them inside, they sleep in our bed with us, they sleep on the couch with us. So we have about 550 dog bloods, and this is a random pooling of dog bloods. These aren't dogs that came in sick, these aren't dogs that were tested for Lyme, it's just a random sampling. So out of the 362 that we've tested so far, almost 35% of them have tested positive for Borrelia. So this is a pretty high number, and it does have some implications for the possibility that household pets, I'm not saying get rid of your pets, absolutely, they're wonderful, but it is important to check your dog and check yourself. You know, even if you haven't been outside in a while, still take a look, because if your dog's been outside, you've been outside too. And then the last thing we have, you might not be able to read this, I know it's kind of small. We do have copies of this paper up at the BU Lyme table if you are interested in it. But we wanted to see what role that deer played in the spread of the causative agent of Lyme. So we were looking at ticks on deer, and the infectivity ranged from 1 to 7%, which is very low. So then we were looking at the ticks that we dragged in the same areas from which these deer came from, and the ticks off the ground, 38 to 55% were infected. So it's very, very different. As it turns out, deer actually have a prophylactic immune response. So the blood of a deer, if a tick that has the Lyme pathogen feeds on deer blood, it will clear the pathogen. But, there's always a but. We also did look at attached versus unattached ticks. So the attached ticks, which was really interesting, 0 ticks were infected that were actively feeding on a deer. The ticks that were unattached, the infectivity was still much, much lower than what it was when we picked them off the ground. So we think that may be due to intermittent feeding before they kind of really attach and go for gold. But deer still do play an indirect role. I'm not saying deer are not a threat, because those of you that are hunters or just have deer in your yard, you know that deer are covered in ticks. As I said before, they're the primary breeding site. So indirectly, they do play a role. They walk them into your yard, they drop them in your garden, and then the adults lay 1,000 eggs in your yard. So indirectly, they do play, but directly, they do not have much of a role in the spread of the pathogen. So I want to thank our research team. This is not even all of them. We have a ton of undergrads and a ton of graduate students. Some of them are in here today. They have posters up in this link hallway here if you want to see what they've been doing on this project. And I also wanted to take just a second to talk about where we're going this summer. So we've been doing the 6-county range within these parks that people often use for 2 years now. So what we want to do this summer is kind of switch gears a little bit. And I've talked to some of you this morning already. We want to go into neighborhoods. We want to see what's happening in your neighborhoods. You know, you kind of say, you know, my backyard is a hot spot, or this elementary school is a hot spot. So if anybody knows of areas, we're in this 6-county range, we're looking at all 6 counties. If you know neighborhoods that are kind of littered with ticks, we would like to know about it. This will help with kind of the public health angle too, to say, look, people live right here, this is what's happening, to kind of get the ball rolling with things. So if anybody has any ideas, again, the BU table is right by that stairwell back there. So if you want to give me an email, give me a contact address, anything like that, we would be very grateful for that. So thank you.